This will be the first lecture of a series of 3 on the gastrointestinal system, and we will briefly review the functional anatomy of the gastrointestinal system. So in this lecture, we will not only review the anatomy of the system, but briefly discuss the functional significance of each of the components. So first of all, there are 4 basic digestive processes. These are motility and secretion, which of course are necessary to produce the functions of digestion and absorption. So motility is of course the actual contraction of the smooth muscle within the GI tract that propels food from the mouth ultimately all the way to the anus. And it is of course assisted by the secretion of digestive enzymes that chemically help to digest the food, in addition to the motility, which is not only propelling food, but also helping to mechanically grind the food. So motility and secretion together, of course, are aiding the process of digestion. Excuse me. In addition, once the food has been digested, it needs to get, of course, absorbed into the GI tract. Into the bloodstream. Now, one of the things that is very important that you highlight right away is the fact that material being absorbed, nutrients being absorbed into the bloodstream, first of all go to the liver, where they are processed before entering the general circulation. So the mouth, pharynx, esophagus, and stomach are the first half of the GI system. Of course, in the mouth, food is chewed, masticated, etc., swallowed, going down through the esophagus into the stomach. Now, here we have a blow-up of a segment of the wall of the stomach. We have these gastric pits, which are openings of glands that are in what we call the gastric mucosa. These glands consist of chief cells that secrete pepsinogen, and we'll be talking about these different enzymes, digestive enzymes, in the second segment of this series, as well as parietal cells that secrete acid. And these are all part of what we refer to as the gastric glands. So these, of course, are all involved in secretion, which is, of course, involved with the process of digestion. So the mouth is where digestion begins with a process of mastication, technical term for what we refer to as chewing. It's also secretions, saliva, which is a lubricant. It aids in the swallowing of the food material down through the esophagus. Lysozyme, which is an enzyme that kills bacteria. Salivary amylase, which digests starch and glycogen. Remember, starch is the animal form of stored carbohydrates, whereas glycogen is the Starch is the plant form of stored carbohydrates, whereas glycogen is the animal form of stored carbohydrates. And salivary amylase begins the process of digestion by breaking down these substances, as well as other more simple carbohydrates that are in the foodstuff. Of course, then material is swallowed, it goes into the stomach. The stomach can stretch quite a bit. Its empty volume is down around 50 milliliters, whereas when it's packed full of food, it can expand to as much as 1,000 milliliters. It secretes gastric juice. It also has contractions, which help to mix the chyme, that is the ground up bits of food coming down from the mouth, as well as mix that chyme with the gastric juices. So these two, of course, processes are related. The contractions also help gastric emptying down into the small intestine. And food is released into the small intestine slowly, bit by bit, small quantities. So in the stomach, first of all, as you said, it expands to accommodate food as it's swallowed. The term rugae is used to refer to these folds that flatten on expansion. We can see those a little bit better in this previous slide here. Then the gastric pits, which I've referred to in a previous slide, these are located in the stomach lining, And the pits actually are the opening of the necks of the gastric glands. And they have several secretory products. Pepsinogen, which is secreted by chief cells. It's a precursor for pepsin, which is the actual digestive enzyme. One thing you need to keep in mind is that because these enzymes can actually degrade protein, and of course the stomach itself, as well as the rest of the GI tract, is made of protein, that you need to release an enzyme or a proenzyme, a precursor to the enzyme, that is not capable of digesting the gland itself before it's secreted. Because that, of course, would be a very concentrated environment in which a digestive enzyme would do a very nice job of destroying the very gland that produced it. So we have these precursors, such as pepsinogen. Also, there are hydrogen ions that are secreted by parietal cells, and these are going to be producing the hydrochloric acid, that is not only helping to digest food, but it's also going to activate pepsinogen into pepsin. We'll talk about that more later. But its main function, of course, is preserving the acidic environment of the stomach. Intrinsic factor, which is secreted by parietal cells. Intrinsic factor is necessary for the absorption of vitamin B12. Gastrin, which is secreted by G cells as a hormone. We'll talk about its role in regulation of digestion later on. And mucus, which is secreted from the neck cells, that is, cells that are actually in the neck of the gland here. And this is serving somewhat of a lubricating function, but also ends up coating the surface of the stomach lining, which is going to help to protect it from the digestive processes that are going on nearby. So we've mentioned the acidic environment of the stomach. The pH in the stomach is 2. Now keep in mind that normal blood pH is 7.4. So this is distinctly more acidic than the blood. And this is necessary for activating pepsinogen, as I mentioned before. It also denatures proteins. So from that standpoint, it is helping to digest proteins. It helps to kill bacteria. And then, of course, we have to have, as I said, this mucosal barrier, which produces a protective layer of mucus and bicarbonate. So the mucus and bicarbonate, which, of course, are going to be right on the surface of the lining of the stomach. The mucus tends to act as a physical barrier that helps to stop the aqueous environment, which contains the acid, from getting to the underlying cells and damaging them, as well as the bicarbonate, because bicarbonate, of course, being an alkaline substance, is going to help to neutralize acid. So this, again, helps to produce a protective lining right at the surface of the stomach that helps to protect it from being digested. And this mucus and bicarbonate are secreted from the neck cells and the goblet cells, which are located, as the name suggests, in the neck of the gastric glands. Now, the stomach empties food into the small intestine through the pyloric sphincter. So it regulates passage of chyme between the stomach and small intestine. This is important because we want the small intestine to not be overwhelmed with material that needs to be absorbed, because most of the absorption in the GI tract occurs in the small intestine. So we need to be able to regulate the amount of material going into the small intestine so that the supply does not exceed its ability to be absorbed. So the small intestine is a coiled hollow tube. It's actually fairly extensive. It's about 8 to 10 feet long. And of course, it lies between the stomach and the large intestine. One reason it's so long is it helps to provide an enormous surface area, and that's important for absorption of nutrients. It is a primary site of continuation of digestive processes, which of course starts in the mouth, continues in the stomach, and is finalized in the small intestine, as well as absorption. Now, almost all of the absorption of nutrients occurs within the small intestine, and most of that absorption is completed within the first 20% of the intestinal length. The small intestine is anatomically arranged to produce a large surface area for absorption. So it's not just the length of it, but also it has a lot of convolutions, what we call the brush border. We'll talk about that a little bit later, but many different projections, what we call villi, that extend into the lumen of the small intestine that help to increase the surface area even more. Small intestine is divided into three divisions: the duodenum, the jejunum, and then the ileum. So most of the absorption is occurring within the duodenum. Therefore, there's a fair amount of secretions into the duodenum to help finish the digestive process. Pancreatic juices. This contains more digestive enzymes, as well as bicarbonate. Now, in this case, the bicarbonate is even more important than it was in the stomach because we've got the chyme coming in, which is highly acidic. And in order to neutralize that acidity, this bicarbonate is released to help produce the small intestine. The small intestine, the lining of it is not at all well designed for protection otherwise without this bicarbonate. So we really need the bicarbonate to help protect the tissues from digestion. Also bile. Bile is secreted from the liver. It can be stored in the gallbladder, and it contains bile salts, which aid in fat digestion. And then we'll talk about bile salts and how they function in fat digestion in the second segment of the GI lectures. Let's take a look at the wall of the small intestine. As I said, it has many, many villi that it project into the lumen, and they're shown in aggregate here. This shows you an individual villus, and not only do these project into the lumen, but the cells on them have many microvilli that increase the surface area even more. So, of course, this is very important from the standpoint of enhancing absorption. You need an enormous surface area. Within the villi themselves, or the mucosa, you have arterial blood supply, venous blood supply, as well as shown here in green, the lacteal. The lacteal is a lymphatic vessel. We mentioned them briefly when we talked about the immune system. And the lymphatic is important for two different ways. If you consider the fact that the material coming down through the lumen here in the digestive tract is coming from the environment. So it potentially contains many harmful substances, as well as obviously the useful substances, that is, nutrients. So it makes sense that we have a lymphatic system present to help identify the presence of any foreign substances that may be harmful and to deal with them, to permit the immune system to have close proximity to deal with them, as well as the lymphatics are the primary vessel that will be involved in the absorption of fats. And we'll see that again in the second installment of the GI lectures. So, as I said, the villi increase the surface area of the epithelium and contain these blood vessels in the lacteal. Microvilli increase the surface area even more, and they form what we call the brush border. And there are many enzymes located in the brush border, and these are called, because of that, brush border enzymes. There are also the crypts of Langerhans, and these are epithelial cells in the crypts that secrete a bicarbonate-rich fluid. I've mentioned this in a previous slide. And this is secreted in the proximal portion of the small intestine, so largely in the duodenum. But the bicarbonate is recirculated, so it's actually absorbed in the distal portions of the small intestine and then reused. The liver plays an important role in digestion, especially absorption. All absorbed nutrients travel in the blood to the liver first, before entering the general circulation. So we have extensive vascularization going to both the small intestine and even to the large intestine that are going to aid in this collection and absorption of materials. And the liver functions to detoxify substances and process certain nutrients. So the liver, first of all, produces many different enzymes that our body uses, and some of these are for detoxification, and some of them are used in processing of nutrients, as well as the liver plays an enormous role in the absorption, processing, and even synthesis of lipids. And we'll talk about that later on. So for the liver to get this materials from the GI tract, we have the hepatic, what's called the hepatic portal system. And this is the vasculature that delivers these absorbed nutrients to the liver. Excuse me. So the nutrients absorbed from the small intestine enter into the mesenteric veins. These are what we see here, and travel from the mesenteric veins to the liver via the hepatic portal vein. And then, of course, after being processed within the liver, they travel through the vena cava into the general circulation through the hepatic vein. The hepatic artery provides the liver with fresh blood to supply oxygen. So the hepatic artery is helping to keep the liver alive. And in that context, the liver is helping to keep the body alive by both detoxifying harmful absorbed substances as well as processing nutrients. The large intestine is probably functionally the simplest part of the digestive tract. First of all, it consists of three sections: the cecum, the colon, and the rectum. It has an ileocecal sphincter. This is misspelled here. There should be an H in there. Ileocecal sphincter, located between the ileum and the colon. And then what are called tenei coli, that are bands of longitudinal smooth muscle that surround the large intestine and give it this characteristic bulging kind of structure. The large intestine is largely, its main function is to finish absorbing water out of the chyme so that as much water is conserved as possible, and also to help dehydrate and compact the fecal matter, as well as to store it until defecation. So basically the colon is a hollow tube. It goes from the small intestine to the rectum. And as I said, it concentrates the waste product into feces, absorption of a great deal of water. Any of the water that was not absorbed in the small intestine is going to be stored there, and then of course it's storing the feces until the process of defecation. The rectum and anus. The colon empties into the rectum, and then the feces are excreted through the anus. There is an internal anal sphincter, which is smooth muscle under autonomic control, which means, of course, that it is not capable of being consciously controlled. So it responds to the stretch or lack of stretch within the rectum. The second sphincter, which is closer to the exterior, is the external anal sphincter, and this is skeletal muscle. And because it's skeletal muscle, it's under voluntary control. This is the muscle that infants need to learn to control in order to have undergone successful potty training. Relaxation of both sphincters is necessary to allow the process of defecation. Now, throughout the digestive tract, we have a series of accessory glands. Basically, these are responsible for secretion of products via ducts into the lumen of the GI tract. And these include the following: the salivary glands, which secrete saliva, the pancreas, which secretes pancreatic juice, and the liver, which secretes bile. And of course, this also means that these are secondary endocrine organs, because they are involved in secretion of various substances, some of which are in fact endocrine and some are exocrine in function. So let's take a look at the structure of accessory glands. Here we have the body of a gland attached to ducts that are going to enter into the lumen of the digestive tract. These glands release what we call a primary secretion, which in most cases is a proenzyme, or prohormone in some cases, meaning that it does not actually have enzymatic activity, but that it needs to be modified. And the modification is going to eventually lead to the production of the active enzyme or active hormone. And the modification of it is in large part due to transport of ions and water changing the chemistry of this primary secretion. The glands are secreted by epithelial cells, which of course, some of which are involved in the synthesis of the substances, and some are involved in just providing a protective surface. So saliva, one of these accessory glands, first of all, is rich in bicarbonate. The bicarbonate is itself, because it's alkaline, is going to facilitate some digestion. Also contains mucus, which is to help lubricate the material so that it can be swallowed more easily. Contains some enzymes, salivary amylase, which breaks down starch and glycogen. So this is where digestion begins, of course, in the mouth, and it is due exclusively to the initial breakdown of carbohydrates, the starches and glycogen. And then lysozyme, which kills bacteria. So also helps to protect in serving a minor immune function. Another accessory gland is a pancreas, which has both exocrine and endocrine portions. The endocrine portion, what we refer to quite literally as the endocrine pancreas, releases insulin, which of course is important for the absorption of glucose by all cells in the body. And we'll be talking about the function of insulin more thoroughly when we get into the next chapter, where we talk about energy regulation. The exocrine portion of the pancreas produces what we call pancreatic juice, which also is rich in bicarbonate. As I said in our previous slide, this helps to protect the lining of the small intestine from the highly acidic chyme that's coming in from the stomach. It contains other enzymes, pancreatic amylase, and amylase always breaks down starches and glycogen, as well as lipases, which break down lipids or fats. The pancreatic juice also contains proteases that break down proteins and nucleases that break down nucleic acids, which are derived from proteins. So digestion begins in the mouth with the digestion or the initial breakdown of carbohydrates, which continues in the stomach. The stomach starts to break down proteins, and we complete the digestion of carbohydrates in the small intestine with the pancreatic juices, and we continue the breakdown of proteins in the small intestine because of the pancreatic juices. And this is where the breakdown of lipids really begins as well. Now, I mentioned before the idea that these pancreatic enzymes, not just pancreatic, but digestive enzymes, have to be released in an inactive form to protect the digestive glands from digesting themselves. And we refer to these substances as zymogens. Zymogens are the inactive forms of digestive enzymes. They are stored in what's called zymogen granules of acinar cells that are in the mucosal lining of the GI tract. And they are activated usually by proteolytic enzymes. So these are enzymes that do break down proteins, but are not powerful enough to act as digestive proteins. And these proteins are themselves located in the lumen of the digestive tract. And we'll talk in more detail in the next handout about which enzymes break down which zymogens. The biliary system, trying to get the slide to load here. The biliary system, as the name suggests, is involved in the production and release of bile. And it consists of the liver, gallbladder, and associated ducts. So the liver synthesizes bile, the gallbladder stores it, and the bile leaves the gallbladder through the common bile duct, which transports bile from the liver and or gallbladder down to the duodenum. And the function of bile is to break down fats through the process that we call emulsification. And we'll talk about this in more detail in the next lecture. The common bile duct joins with the pancreatic duct to form what's called the ampulla of Vater. And the drainage of the ampulla of Vater is controlled by the sphincter of Oddi, which regulates the flow from the pancreas and gallbladder into the duodenum. We've talked a little bit about the liver before. I would talk about it in a bit more detail here. First of all, it secretes synthesizes and secretes bile. Mentioned previously that it processes nutrients. Glucose is converted into glycogen. Now keep in mind that glycogen is a storage form for glucose. Just as I said in plants, starch is a storage form for carbohydrates, primarily glucose. In animal tissues, it is glycogen. So the liver is involved in storage of carbohydrates, primarily conversion of glucose into glycogen, so that it can be used after we eat. When we talk about regulation of energy balance, we'll talk about two phases of basically the digestive process or energy balance process. And these are the absorptive phase, which is primarily what we're discussing in this chapter on the GI tract. That is the absorption of nutrients. And then what's called the post-absorptive phase. Now, the post-absorptive phase is greater than 2 hours after you eat a meal. So in that case, the glycogen is then going to be broken down back into glucose. But during the absorptive phase, we're not only absorbing glucose and using it, but we're also converting whatever is not immediately used into glycogen. So we have this process that swings back and forth between the absorptive and the post-absorptive phase, and that's why we have this sort of a process going on in terms of glucose being stored as glycogen, which will ultimately be converted back into glucose. Liver also converts amino acids into fatty acids, which again serve as a storage form of energy, of both, in this case, some protein as well as lipids. Triglycerides and cholesterol are synthesized by the liver from these fatty acids. Liver is also involved in lipoprotein synthesis. Liver also removes old red blood cells from the circulation, so it breaks down hemoglobin and from that generates bilirubin. And is also involved in elimination of waste and toxin. Bilirubin, for example, is eliminated as bile pigments. Also, liver synthesizes plasma proteins, various globulins that we've talked about, and secretion and modification of various hormones.